Good afternoon. I would like to thank the organizer for this opportunity to present our work on complex genomic rearrangement signatures in cancer. Genomic rearrangements are also called structural variations. They are large-scale structure change of the DNA in the genome. They include deletions, insertions, inversions, duplications, translocations, and other complex forms. For example, if the red fragment of the reference genome is removed, it's a deletion. If it's flipped in orientation, it's an inversion. Speaking of complex rearrangements, chromothripsis is an extreme case. This is one example of a kidney cancer genome. The chromosome 1 to chromosome Y are listed in a circle. These colorful lines in the middle are individual somatic rearrangements specific to cancer cells. So this kind of phenomenon argues against the common knowledge about mutations accumulate slowly over time and one or two events per cell cycle, and over decades, one of the normal cells acquire enough mutations and turn into a cancer cell. Chromothripsis can only occur as a single event because there's no such mechanism that we know of that could target one or two chromosomes over and over to form this kind of pattern, while leaving other chromosomes alone. And later on, there are experimental evidence showing chromothripsis that indeed occur through one cell cycle. In case you never heard of the word chromothripsis, one day I was watching an animation movie with my kids, Spies in Disguise. This guy here was a spy. This boy was a scientist who made a drug supposed to turn human invisible. Then the spy drank that drug. Then his genome was undergoing chromothripsis. When I watched here, I was so impressed by the Hollywood screenwriters that they are so up-to-date to science. To detect this kind of complex rearrangements from whole genome sequencing data, our colleague Isidro Siriano-Cortez developed an algorithm called HiTSeq. Basically, it's looking for a pattern like this: a lot of rearrangements on one or a few chromosomes with crossing pattern of the breakpoints. And individual rearrangement, deletion, duplication, inversions are equally represented, unlike the bottom two can be explained by individual SVs accumulating over time. So applying this algorithm to 2,400 whole genome sequencing tumors from 37 tumor types in pan-cancer analysis of whole genome consortium, we detect about 2,000 complex rearrangements in about half of the tumors in this cohort. There are several mechanisms known to produce chromothripsis in cells. For example, micronuclei and the chromatin bridge are two aberrant cellular structures formed during error of cell cycle. In micronuclei, during cell division, different chromosomes get pulled towards two different daughter cells, and sometimes several chromosomes will be lagging behind. When the nuclear envelope forms, most of the chromosomes will form main nuclei, but the lagging chromosomes will form micronuclei. In micronuclei, DNA replication and the DNA damage repair are deficient, and the chromosomes in micronuclei sometimes will shatter into many small fragments, and some fragments get lost. Some are randomly stitched together into a new chromosome, and it's been shown under microscope that the DNA can really break down into many pieces. And in chromatin bridge, it starts with a dicentric chromosome. During the cell cycle, the two different centromeres get pulled towards two different daughter cells and forming a fiber connecting two cells. And the fiber bridge is visible even several hours after cell division. So this DNA bridge can break into, again, many pieces and randomly stitch together. And the broken piece can sometimes ligate into another chromosome, form another dicentric chromosome, and this cycle can go on for several rounds. So in micronuclei, the DNA damage seems to be random across the chromosome, so the resulting chromothripsis will demonstrate an even distribution of breakpoints. And in chromatin bridge, the DNA damage is not random, so you would observe several hotspots of rearrangement breakpoints. And then it's also been shown that circular extra chromosome DNA can come from complex rearrangement. Non-negative matrix factorization-based approaches have been widely used to study the mutation signatures and the molecular mechanisms of point mutations. And the derived signatures often reflect the molecular mechanisms very well. And we try this type of approach a lot, trying different ways of constructing input matrix, and the decomposed signatures are often not very satisfactory. In the end, we realized this is probably not the best approach, because for complex rearrangement, each tumor there are usually only one or two such events, but for point mutations, there are usually a lot. So instead, we developed an algorithm called Starfish, using a very different approach. This was done by a talented postdoc Li Suibao in the lab. So it's very simple that we use 5 different features to describe the overall patterns of the copy number profile and the breakpoint distribution of individual complex rearrangement events. So these features include breakpoint dispersion score, which describe the randomness of breakpoints across chromosome, and also include amount of copy loss, copy gain, the maximum copy number, and the amount of telomere loss. Then we ask a simple question. What if we put complex rearrangement events that look similar to each other together? So we performed unsupervised clustering analysis, and we found there were 6 clusters. And now we referred these to 6 signatures. Now we expect some of these signatures would correspond to known molecular mechanisms. We collected 5 published studies. All of them have induced chromosynthesis in cell culture systems by inducing either micronuclei or chromatin bridge. And then we paint the colors of these events by the 6 signatures, so we could see the micronuclei-induced chromosynthesis are painted mainly by this light green color, and the chromatin bridge-induced chromosynthesis are mainly in orange color. Now we come back to this figure of signatures. Signature 4 is the light green signature, which is micronuclei. It has the lowest breakpoint dispersion score, which means the breakpoints are evenly distributed across the chromosome, which is consistent with what we know about micronuclei-induced chromosynthesis. And the orange signature is signature 2. It has the highest amount of telomere loss, which is also consistent with chromatin bridge-induced chromosynthesis. Here, I'm showing you a few examples. This is the same kidney cancer genome that I showed you earlier, which is predicted to be signature 4 micronuclei. So the breakpoints here are evenly distributed across the chromosomes, and the copy number profile shows the copy number oscillating from 2 to 3 state. And this example is a liver cancer genome predicted to be a signature 2 chromatin bridge that you could see. The breakpoints are highly concentrated in very small regions, accompanied by very large fragments from the telomere side is lost. Signature 1, we believe, is associated with extrachromosomal DNA, which is a small circular form of DNA molecules that are usually highly amplified in cancer. This one is an example of leiomyosarcoma genome that you can see a lot of small fragments are highly amplified into dozens of copies. Now, out of these 6 signatures, we are able to connect at least 3 signatures, signature 1, signature 2, and signature 4, with known molecular mechanisms or biological processes. There are 3 other signatures we cannot connect to known processes. One we named large loss, another large gain, showing here that the features, they have a large amount of copy loss and copy gain. And signature 6 is very interesting. We name it hourglass chromatosis. The main feature is it has the highest breakpoint dispersion score, meaning the breakpoints are highly localized, and there's almost no copy gain with a very small amount of copy loss. Here is an example of hourglass chromatosis. You could see that the rearrangement breakpoints are concentrated in a few regions, and every time you have rearrangement, there's a small amount of DNA loss. Now we can take a look at how different signatures distribute across different tumor types. Here, each block of colors represents one tumor type scaled from 0 to 100%. And each individual horizontal bar represents one tumor. And if there are multiple colors horizontally, that means one tumor carries multiple complex rearrangements by different signatures. So overall, we have glioblastoma, which is a brain cancer, sarcoma, esophageal cancer. They have the most abundant complex rearrangement, up to 80%. And on this side, pediatric brain cancer, CLL, leukemia, and thyroid cancer, they have the least number of complex rearrangements. And also you see a bias in different colors. For example, sarcoma and GBM, they have abundant ecDNA, which is consistent to what we know about these tumors. And in kidney cancer, they have a lot of chromatin bridge-based chromatosis, which is also consistent with what we know. And surprisingly, this signature 6 hourglass chromatosis I showed you earlier, they are so abundant in prostate cancer. About half of the prostate cancers carry hourglass chromatosis. Then we collected a total of 500 whole genome sequenced prostate cancers from these 4 different cohorts, and we found the mutations in this one particular gene called SPOP is associated with 3 different signatures of complex rearrangement. Signature 6, hourglass, signature 4, micronuclei, and signature 3, large loss. SPOP is a E3 ubiquitin ligase. The normal function is to bind its target and degrade them. And the mutations occur in 6% of the prostate cancers, and they are mutually exclusive with ETS fusions and a PTEN loss. All these mutations are missense mutations in one particular domain, which is a target binding domain. So our results suggest the wild-type SPOP protein may play some important role in maintaining genome stability, and the mutant SPOP is promoting genome instability. Another interesting thing we found is related to transcription and the DNA replication conflict. Since DNA replication and the transcription, they both use the same DNA as a template, it is possible for the DNA polymerase complex and RNA polymerase complex to run into each other and cause a crash. And based on the relative orientation, we could divide them into a head-on collision and a co-directional collision. Even if the 2 polymerases goes to the same direction, it is still possible for them to crash because the 2 polymerases travel at different speeds. Then we could use replication timing profile produced by RepliSeq experiment. In these experiments, these regions are early replicated regions, and these bottoms are late replicated regions. And then we could define left replicated region and a right replicated region based on these sharp transition regions in these profiles. Then for every gene, we could define them as a head-on collision gene or co-directional collision genes. Then we test the enrichment of the rearrangement breakpoint belongs to these 6 different signatures. And we found ecDNA breakpoints are enriched in head-on collision regions. So if the head-on collision is causal to produce ecDNA rearrangement, we would expect this kind of enrichment depends on gene expression, because if there's no expression, then there's no transcription, then there's no collision. So then we divide all the genes based on their expression level. So in top 50% of the genes by expression, we observe the enrichment, but not in the bottom 50% of the genes by expression. So this is consistent with a model we proposed that the DNA polymerase, RNA polymerase crash, and the replication fork would collapse, and the DNA polymerase sometimes would jump to another strand, which is called template switching. This is a known mechanism that exists in cells, and if this jump occurs again and again, it can produce complex rearrangement. So to summarize, I've shown you that complex rearrangements is very common in cancer, and we detect 6 different signatures. ecDNA is associated with transcription replication conflict. Our glass chrome thripsis is a new type of complex rearrangement enriched in prostate cancer. SPOP mutations are associated with 3 different signatures, and they are related to elevated genomic stability. And our manuscript on this study has been accepted by Nature Cancer. They should be online soon. Thank you for your attention. Again, the work is done by a postdoc in the lab, Lixing Yang, who is currently associate professor in China. These are the funding agencies. I'm happy to take questions.